Hello, everyone. Today I'll walk you through the quantification workflows using Triple Quadrupole Mass Spectrometry. In many scientific fields, detecting a molecule is not enough. We often need to know exactly how much of that molecule is present in a complex sample. This could be a drug in human blood, a biomarker in clinical samples, or a pesticide in food. Triple Quadrupole Mass Spectrometry has become one of the most reliable technologies for achieving highly accurate and sensitive quantitative measurements. In this presentation, we will explore how this technology works and how scientists use it in a structured workflow, from sample preparation to final validation of results. Let's begin by understanding why quantification is so important in analytical science. In drug development, researchers need accurate pharmacokinetic data to understand how a drug behaves inside the body. Quantification helps determine safe dosage levels and therapeutic effectiveness. In clinical diagnostics, scientists measure biomarkers in blood or tissue samples to detect diseases early and monitor treatment responses. In food safety and environmental monitoring, analytical laboratories must detect contaminants like pesticides, heavy metals, or pollutants, often at extremely low concentrations, sometimes at parts per billion levels. These applications require analytical techniques that are highly sensitive, selective, and reproducible, which is exactly where Triple Quadrupole Mass Spectrometry excels. A triple quadrupole mass spectrometer consists of 3 quadrupole analyzers arranged sequentially, commonly referred to as Q1, Q2, and Q3. The first quadrupole, Q1, acts as a mass filter. It selects ions based on their mass-to-charge ratio, isolating the precursor ion of interest. The second quadrupole, Q2, is called the collision cell. Here, the selected ions collide with an inert gas such as nitrogen or argon, which causes the ions to fragment into smaller product ions. Finally, the third quadrupole, Q3, filters these fragment ions and allows only specific product ions to reach the detector. This sequential filtering process greatly enhances specificity and sensitivity, which makes triple quadrupole instruments ideal for quantitative analysis. The key analytical technique used in triple quadrupole instruments is called multiple reaction monitoring, or MRM. In MRM mode, the instrument monitors a specific transition between a precursor ion and a product ion. First, Q1 selects the precursor ion corresponding to the molecule of interest. Next, fragmentation occurs in the collision cell. Finally, Q3 detects a specific fragment ion generated from that precursor. By monitoring this precursor-to-product transition, the instrument ensures that the detected signal truly belongs to the target molecule. This approach provides very high selectivity, dramatically reduces background noise from other compounds, and enables detection limits down to subparts per billion levels, even in highly complex samples. Every quantitative workflow begins with careful sample preparation, which is critical for obtaining reliable analytical results. Real-world samples such as plasma, food extracts, or environmental samples contain many interfering compounds. Therefore, analysts perform extraction techniques such as protein precipitation, solid-phase extraction, or liquid-liquid extraction to isolate the analyte from the sample matrix. Next comes cleanup, where interfering components like lipids, salts, and other matrix substances are removed to improve measurement accuracy. An important step in this process is the addition of internal standards, often isotope-labeled compounds that behave similarly to the analyte. These standards help correct for variability during extraction, ionization, and instrument analysis. Once the sample preparation procedure is established, the next step is method development. First, the analyte is infused into the mass spectrometer to identify its precursor ion, typically the protonated molecule represented as M plus H+. Next, collision energy in the collision cell is optimized to produce stable and reproducible fragment ions. From these fragments, analysts select specific MRM transitions that provide strong signals and good selectivity. Typically, 2 transitions are chosen. One for quantification, known as the quantifier ion, and another for confirmation, known as the qualifier ion. This optimization step ensures that the analytical method is both sensitive and highly specific. The next stage of the workflow is establishing a calibration curve, which forms the basis of quantitative measurement. A series of standard solutions containing known concentrations of the analyte are prepared and analyzed using the developed method. The instrument response, usually the peak area ratio between the analyte and internal standard, is plotted against the concentration. This relationship is typically described by a linear equation, expressed as y equals mx plus c. For a reliable quantitative method, the calibration curve should show strong linearity, often with an r-squared value around 0.999, while accuracy and precision remain within acceptable limits, typically within plus or minus 15%. Once the calibration curve is established, unknown sample concentrations can be calculated. After calibration, the actual sample analysis begins. Most workflows combine liquid chromatography with mass spectrometry, commonly known as LC-MS. The liquid chromatography system separates compounds present in the sample before they enter the mass spectrometer. This step reduces interference and improves detection accuracy. Once separated, the compounds enter the Triple Quadrupole instrument, where the MRM transitions are monitored automatically. The instrument generates chromatographic peaks corresponding to the analyte and internal standard. The area under these peaks is measured through peak integration. Using the calibration curve, the instrument software calculates the exact concentration of the analyte in each sample. Before the method can be used routinely, it must undergo quality control and validation. Several parameters are evaluated to ensure the method produces reliable results. Accuracy measures how close the measured value is to the true value, typically within plus or minus 15% of the nominal concentration. Precision evaluates reproducibility and is expressed as relative standard deviation, which should generally be 15% or less. Another important parameter is the limit of quantification, or LOQ, which represents the lowest concentration that can be reliably measured. This usually requires a signal-to-noise ratio of at least 10. Meeting these criteria ensures the method is scientifically robust and suitable for regulatory applications. To summarize, Triple Quadrupole Mass Spectrometry has become a gold standard for quantitative molecular analysis. Its high sensitivity allows detection of molecules at extremely low concentrations, even within complex biological or environmental samples. Its high specificity comes from multiple reaction monitoring, which ensures that only the target analyte is measured. By combining careful sample preparation, optimized method development, calibration, and validation, scientists can obtain highly accurate and reproducible quantitative data. Because of these advantages, triple quadrupole instruments are widely used in pharmaceutical research, clinical diagnostics, food safety, and environmental monitoring. Thank you for your attention.